Thank you very much for the opportunity given to me to give a talk on this. I have been in this field for last 30 plus years, and I was trained at the Hormel Institute by Dr. Harold Schmid, who is now retired, but an expert in ether lipids and anandamides, trained by Wolfgang Baumann and Hermann Schlenk, Ralph T. Holman, and these are legends in lipidology. Actually, this is, let me be honest with you, it's old wine put in new bottle. It's lipidology. That's it. I'm a lipid chemist, and also I'm an enzymologist dealing with the lipidology. I love the field. Life as a matter of fact. That's it. And I thank the CCTS organization, Mike, and others, and my division, Pulmonary and Critical Care, Department of Medicine, and the Pharmacology, College of Pharmacy, and also my institute, Davis Heart and Lung Institute, for supporting me. A lot of work I did here, actually I grew up here without a lot of funding. And actually they put up with me, and we published about 70 papers in the last 6 years in the field, last 7 years. And we have people I have to thank, Rao Madhipati at Wayne State, where he has a center for lipidomics, and we are actually working with him. Andrew Morris, who actually cloned the PLD gene. He is at the University of Kentucky. He supports us. Viswanathan Natarajan was my mentor at Chicago, and he's also a lipidologist. And Dr. Eric Kraut, we have just gotten into the sickle cell disease, the lipidomics of sickle cell disease, and we are working on it. And thanks to Dr. Kraut for actually inviting me into the project. This is what it is. These are different species. There are about, according to the lipid maps, there are about 1,080 lipid species in the cell. And the flux is so much, even for an analytical lipidologist, it is so difficult to do that, to do the complete analysis of these molecules at a metabolome level. And fatty acids, or the precursors for those, all these complex lipid molecules. And what are the functions of these lipids? There are several functions. One of these, the energy storage. All this talk about triacylglycerol, triglycerides, the fat, the obesity, all these problems are because of the storage. Structural elements is an old terminology. There is nothing like a structural element for a lipid. Every lipid that is a structural element is a functional element. And hormones, most of the sex hormones, other steroid hormones, they are derived from the steroids, and that's why cholesterol is very important. And the enzyme cofactors, there are several lipid enzyme cofactors, lipidic enzyme cofactors. They're highly functional and electron carriers, and also the pigments in the plants, and emulsifying agents. All your bile salts are also lipidic. They are actually cholesterol-based. Actually, they are derived from cholesterol. And finally, this is a field actually it exploded in the last 10 to 15 years, is the bioactive lipid signaling molecules. They actually mediate from the membrane till the nucleus and back. And these molecules are very interesting and also very complicated to look at. Fats, oils, greases, and these are all synonyms for these lipids. Lipids is a term that is used commonly in the scientific arena. And these are typically non-polar and hydrophobic compounds. These are not soluble in water, and you have to extract them. You have to actually isolate them, separate them, derivatize them, then analyze them by different techniques. And most of these are hydrocarbons. Actually, they're long-chain hydrocarbons, and they're typically isoprene type of molecule. And then, actually, the fatty acid is nothing but an extension of an isoprene molecule. You often hear about saturated, unsaturated fatty acids, and the saturated fatty acids have no double bonds, and the unsaturated fatty acids have double bonds. All these omega-3 fatty acids and all these things you hear about, or they're multi-polyunsaturated fatty acids with 6, 5 double bonds, and we need those. Okay, let me actually tell you why I'm giving an introduction on these molecules is because this is oftentimes a marginalized subject. And even in the biochemistry class, there will be only one lecture on this, and there will be one question on this probably. And the steroids also, they're also lipidic in nature. Cholesterol is a typical steroid that is actually present in the cells, and even in the circulating lipids, it has gotten a bad name, unfortunately. But the cholesterol in the brain cells, in the neurons, is about 40 to 50% of the lipid is cholesterol in the brain cells. And the triglycerides are actually, the typical name signifies there are 3 fatty acids attached to the glycerol backbone. The adipocyte is the one that stores lipid cell in the body, and this actually stores these triglycerides, and it's an interesting, very crucial cell in the body. And what happens is when these fatty acids are esterified, actually because the fatty acid is a carboxyl terminal is an acidic one and acidifies on the alcohol region of the glycerol, it's a trihydric alcohol and forms a triglyceride. It's very interesting because there are enzymes that actually regulate the triglyceride metabolism. One of the biggest problems that is actually coming up now is the hydrolysis of these triglycerides and releasing free fatty acids, which actually cause insulin resistance. And the other problem is also with obesity and all these things. We actually have a grant on this, and with Dr. Yuli Megalong on sleep apnea, and we are actually studying these triglyceride patterns and how the triglyceride metabolites actually cause obstructive sleep apnea. Now comes the cell membrane. Okay, the reason without a cell membrane, there is no cell. Without a DNA, there is a cell. Red blood cell can survive. But if you take the membranes out, there is no cell. So the cell has chosen to have these lipids as the barrier. It's not just a barrier, it's also a functional boundary for the cell. And these lipids are typically phospholipids. The phospholipids are very similar to the triglycerides, but one of the fatty acids is taken out, and actually a phosphate and a head group are introduced for the polarity of it. And these membrane lipids have both hydrophobic and hydrophilic regions. They have to repel things, and also the fatty acids act as hydrophobic regions so that they can form a nice boundary. And also they have hydrophilic regions like the phosphate and the head group that actually interact with the outside as well as the inside of the cell with the aqueous milieu. These are typical ones. Each phospholipid has 2 fatty acids. And these fatty acids are anywhere out of the 23 fatty acids. You can imagine the permutations and combinations. There are different phospholipids actually. They form these membranes as these typical membranes are called bilayer membranes. About 75 years of research has actually given the Singer-Nicholson's fluid mosaic model. They worked very, very diligently on this membrane model. And this is very typical in both eukaryotes and prokaryotes. So these phospholipid bilayers, what they do is they form a boundary. They actually help in transport. They selectively operate the transport mechanism. They don't allow everything. They selectively allow everything, allow something. So the lipid membranes are fluid in nature, and also they have both saturated and unsaturated fatty acids. Now, why do they have the saturated and unsaturated fatty acids? The unsaturated fatty acids actually give a kink to that. So the fluidity as well as the asymmetry are maintained by these unsaturated fatty acids. If you have more unsaturated fatty acids, you have higher permeability. If you have more saturated fatty acids, you have less permeability. Similarly, most of the gases are dissolved. Oxygen actually is dissolved in the membrane lipids. Nitric oxide is dissolved in membrane lipids. Actually, these membrane lipids act as storage units. Actually, they form the reservoirs, and they allow the gradients from the plasma membrane to the mitochondria. Mitochondrial death does not happen that quickly because there is a reservoir of oxygen in the cell, so the lipids act as the reservoirs. Even for calcium, intracellular calcium is about 2 millimolar. Most of the calcium is actually bound in the acidic phospholipids. And when they are perturbed, the calcium is released and cellular calcium level goes up from 100 nanomolar to millimolar level. Again, cholesterol is an important one. Cholesterol actually maintains the fluidity of the membrane, and actually it gives the rigidity to the membrane. If you take out the cholesterol, the membrane actually is, the functionality of the membrane, both structurally as well as functionally, is gone. So what the lipids do is actually they also organize the receptors and other proteins in those membranes very nicely for the interactions with the ligands and to cell-to-cell interaction. So the integral proteins go through, and also there are enzymes that are maintained in those. This is a typical cell. So is the plasma membrane the only one that is membranous in nature? No, the organelles are also membranous in nature. The mitochondria have their own membranes. Nucleus have their own membranes. They're connected to the Golgi. So the textbook scenario is So there is a cytosol, there is a C, and all these mitochondria and organelles float in there. No, that's not right. They're actually connected through these membranous meshwork inside from nucleus to the cell membrane. So that's what these rafts, the term they always hear is these rafts they do is actually they work as a cargo. So typically there is a protein to lipid ratio for each membrane. That is universally true. So if this protein to lipid ratio, phospholipid to protein ratio is altered, there is always a scenario that is not good. So certain pathological situations, the protein to phospholipid ratio is altered, and that ratio has to be maintained for the functionality of the membrane. These are the membrane phospholipids, again, the glycerophospholipids, and actually there are different classes of them. Each class has got multiple fatty acids, and each of them actually form different species. One of the interesting one is a sphingolipid. It's actually, these sphingolipids are very important. Actually, they act as one of those things is the human blood group substances, and also they do these interactions with other cells and other adjacent molecules. One of the problems with certain genetic disorders is with the sphingolipid disorders because they actually are accumulated in the lysosomes and they are degraded. And this is one of the lipidic, genetically based diseases that people work on sphingolipids. The second one here is the sphingosine 1-phosphate that is actually a bioactive lipid metabolite that is actually now coming up as an important molecule in maintaining the vascular endothelial integrity. So if you look at this, this is a lipidocentric view, and lipids maintain almost all the majority of the cellular functions from secretion onwards to the cell survival through different metabolites and how they actually regulate their metabolome to operate the functions of the biological cell. One of the things we are interested in is these membrane phospholipids are actually in a constant metabolic flux. There are enzymes that actually hydrolyze these fatty acids as well as the head groups of the phospholipids, and then these metabolites are released and they actually operate the cellular functions. If you look at the glycerol backbone, the number one phospholipid, number one position is a fatty acid. This is usually a saturated fatty acid. In the 2 position of the phospholipid, there is unsaturated fatty acid. And in the 3 position, your phosphate and the head group are attached. So according to the glycerol stereochemistry, in the SN1, if the enzyme acts and hydrolyzes the one saturated fatty acid, it's called PLA1 or phospholipase A1. In the 2 position, there is a specific enzyme, that's phospholipase A2, that actually cleaves this unsaturated fatty acid and releases that. That's why it's called phospholipase A2. In the 3rd position here, PLC, it's a phospholipase C that hydrolyzes the head group along with phosphate. This is in the inositol phospholipid metabolism, and that was called phospholipase C. And the 4th one is called phospholipase D, which only cleaves the head group and leaves the phosphatidic acid. These are actually housekeeping enzymes as well as signaling enzymes. And these enzymes maintain a very tight metabolic function. For example, the phospholipase A2 is very crucial for eicosanoid generation, that is the prostaglandins or leukotrienes. It's such a strict regulated way. Unless phospholipase A2 chops off the 2-position unsaturated fatty acid, cyclooxygenases and lipoxygenases cannot act on those. So they require a free unsaturated fatty acid that has to be released from the membrane phospholipid upon the action of phospholipase A2. Then only cyclooxygenase and lipoxygenase will work and form the eicosanoids such as prostaglandins and leukotrienes, which cause pain and other things. So one of the problems with these lipid enzymes, we cannot isolate, especially from the mammalian cells. It is very difficult to isolate these enzymes. Sometimes we isolate, they lose their activity, and there are certain difficulties. Nevertheless, we still work on these enzymes to look at the metabolic function, how they regulate the cellular functions. This is what the phospholipase C does, forms the inositol phospholipids, which actually regulate calcium as well as activation of protein kinase C. And this one is actually activated by the G protein coupled receptors and then goes through and releases the IP3, the inositol triphosphate, as well as diacylglycerol that actually activates protein kinase C, and other signaling mechanisms happen. This is actually a target for certain diseases, But again, the inhibitors are not that well developed. And the phospholipase D is one of the enzymes that we work on. And this one actually generates phosphatidic acid. And the phosphatidic acid is implicated in mitogenesis and other physiological as well as pathophysiological conditions like cytotoxicity and apoptosis. That is the PA, that is phosphatidic acid. It's also a precursor for other phospholipids. So what we did in our earlier work, these are all published, we actually wanted to see whether oxidants actually activate phospholipase D that actually cause cytoskeletal changes in the endothelial barrier dysfunction. Yes, in fact, the phosphatidic acid generated from PLD activation causes the cytoskeletal alterations in endothelial barrier dysfunction. Then we went and looked at, again, vascular endothelium. We looked at the bleomycin that is actually used as a lung fibrotic agent. And then we looked at whether bleomycin actually causes dysfunction in the vascular endothelium that could be a factor for lung fibrosis. Again, the phospholipase D generates phosphatidic acid, which actually causes disturbance in the vascular endothelium. So finally, what we did, we also used other agonists and to look at confirm whether this actually goes through. Again, if you alter the thiol redox, then you again activate the enzyme that actually causes PA generation, which causes the cytotoxicity. Here, what we did, the last year, Dr. Andrew Morris developed a specific inhibitor, and we used the inhibitor, and actually we can attenuate the phospholipase D activation by these agonists or oxidants. So what we wanted to do here, actually we are still interested is how the substrate, the lipid substrate is utilized by these enzymes and generates a bioactive lipid and then causes either a positive action or a negative action, something like a mitogenesis or apoptosis of cell death or cytotoxicity. So we're actually looking at these enzymes, and one of the interesting things is that these enzymes, phospholipase A2, phospholipase D, and phospholipase C, are present in the nucleus also. And we are looking at, they are nuclear enzymes. Are they similar enzymes? They are actually similar antibody reactions, but we don't believe that they could be different. The kinetics is different. Probably the product formation is different. That's what we are interested in, separating the nuclear versus non-nuclear phospholipases and how they regulate the cellular metabolism. Eicosanoids. Eicosanoids is actually, it is eicosatetraenoic acid. It's starting with the fifth position, the double bond. That's why the oxygenated metabolites of eicosatetraenoic acid, that is, in other words, arachidonic acid, which is present in the 2 position of the phospholipid membrane of the cell, they actually form these eicosanoids, and they are nothing but prostaglandins and leukotrienes. And thanks to Bergstrom, Samuelson, and John Wayne, who actually did that, almost all the work on the physiology of these, and as well as synthesis and characterization of these eicosanoids, especially prostaglandins, which has actually since 1970s, or even before that, it has actually in 82, it has revolutionized the field of lipidomics or lipid biology. As I mentioned earlier, if you have phospholipase A2 that actually chops off the phospholipid at the 2 position, and releases arachidonic acid, that is eicosatetraenoic acid, with 20 carbon long and 4 double bonds. And then that is actually converted into either cyclooxygenase metabolites, prostaglandins and thromboxanes, or lipoxygenase metabolites, leukotrienes or heats, or lipoxins. And there is another enzyme, cytochrome P450 epoxygenase, that actually metabolizes arachidonic acid and forms these bioactive molecules of oxidatively transformed arachidonic acid metabolites, which are physiologically functional. And these are what cyclization. The interesting thing with the cyclooxygenase is actually it cyclizes, and as well as it also introduces oxygen, and also acts as a peroxidase. And it's a very fascinating enzyme. There are 2 isoforms, actually 3, COX-1, COX-2, and COX-3. There are selective inhibitors. They say that COX-2 inhibitors, and it is, I am not sure some of them are really good inhibitors for those. Again, one of the drawbacks with the lipid enzymes, there are no good pharmacological inhibitors for most of the enzymes. And actually, they do more of a damage than actually a positive thing. That is one thing we're actually finding out, that statins, which lower cholesterol, completely rattle all the bioactive lipid enzymes. So so when you deplete cholesterol, the other enzymes are activated. So the biological responses of these prostaglandins are inflammatory responses, pain and fever, and they also cause decrease in the gastric acid secretion, and also control several reproductive functions and so on and so forth. One of the things we were interested in is two lipid enzymes in the metabolome talk to each other. So previously we were only interested in the kinases and other things, and we thought, okay, maybe phospholipase A2 talks to phospholipase D. So before even kinetically in a cellular system, if you activate phospholipase A2, it's activated, and then arachidonic acid is released, and eicosanoids like prostaglandins and lipoxygenase metabolites are formed. They then trigger phospholipase D, and the phospholipase D actually makes phosphatic acid. So this connection, actually, this is also published, and this connection, actually, we are interested in how in the lipidome, 2 to 3 lipid metabolizing enzymes talk to each other and control the lipid metabolism. One of the interesting things what came out of our work is that phospholipase A2, which actually generates free arachidonic acid that is converted into eicosanoids, actually controls the interleukin cytokine, inflammatory cytokine secretion. So a very acute inflammatory response, PLA2 is activated and eicosanoids are generated. In the late inflammatory response, the interleukins are actually triggered by those. If you block PLA2 activation, you can block interleukin 8 release. For all this, what you need is analytical platform. Without analysis, still lipidomic or lipid chemistry, in lipidology is still mostly analytical. If you don't have good chemistry support, it's difficult to do. So what extraction, how do you want to extract these? You have to use organic solvents, preferably suitable solvents. Then you have to separate them by chromatography method. Most of the times we even use thin layer chromatography. Thanks to Helmut Mangold, who actually developed at Hormel Institute the TLC method, we use that and separate those. And then we separate them on either high-performance liquid chromatography or then characterize them on mass spectrometry. This is the series of methods. And typically that's what the extraction, then separation, and then the analysis of those. So let me quickly go through what it does is when you separate them on HPLC, the peaks are like this, and then you take them and further analyze them by mass spectrometry. And then you can know each one of them, what are each phospholipid has, and what are the molecular species present. We call them molecular species. What is in the 2 position, 1 position, because that dictates the fluidity of the membranes and also the function. So typically, I don't want to go into the instrumentation. This is the state of the art. You actually use liquid chromatography mass spectrometry. It's called LCMS with quarter poles, and each one, then you first ionize them, then generate daughter ions, then you generate granddaughter, and you can actually, after the ion spray, you can fragment them and you analyze those fragments, and each one will characterize those. But one of the problems with this is there are no good standards. So each molecule, you have how many, like 23 fatty acids and 1 phospholipid, and you can 1, 2 position, then you can actually determine what are the permutations of those. So one day probably it will become, but the libraries will be built, but the difficulty is that we don't have good standards for each one of those molecular species. So let me quickly skip, and within 4 minutes I'll finish this. This is what the translational aspect. Ed Dennis, who actually is heading the lipid maps in California, is also a very good phospholipase A2 biologist, and he has come up with the separation of these phospholipids, and especially the derived eicosanoids. And this is all called Eicosanomics. The reason I'm showing Eicosanomics is because of the clinical relevance to that inflammatory mediation and other things. These eicosanoids are separated with 1 throughput injection. You can actually separate almost all the eicosanoids if you have real good standards for those. And you separate them on LC, and then I analyze them on mass spectrometry. And then if you look at those, this is what they have done. This is a heat map of those. In the animals that are given hyperalgesia, you can take the cerebrospinal fluid as well as the tissue and can look at the formation of these eicosanoids, both cyclooxygenase metabolites and lipoxygenase metabolites, and cytochrome P450 epoxygenase metabolites. One of the interesting things is these are ethanolamides. These are actually anandamide family members, which Harold Schmidt used to work for 40 years. And these are called enacylated lipids. And they are generated during insult, stress. We used to call them stress lipids. They're actually elevated. And these, we think they're adaptive lipids. That's how the anandamides came. And these are actually due to adaptive response. They are formed when there is a tissue injury. Tissue generates these anandamides, and anandamides protect the tissue from the injury. Anyway, in this work that Ed Dennis has gone and generated this, this is actually done. This is a lipidomic analysis, or it's called Eicosanoids profile of the hyperallergesia in the rats. So now we look at back and again look at the cyclooxygenase metabolites. Look at COX, LOX, and cytochrome P450 metabolites. And these are what's, this is the prostanoid ring of those, and they are different. I don't want to go into the nomenclature. And these are different eicosanoids formed from the cyclooxygenase pathway. And these are leukotrienes as well as cystinyl leukotrienes formed from the lipoxygenase pathway. These are also another type of eicosanoids. Then you analyze them. This is what in the translational medicine is. You need to know the substrate. The substrate is converted to the products. And what is the substrate availability from the phospholipid in the 2 position, and how these are actually shunted into different metabolites of eicosanoids. And each one is actually, each arrow is actually a gene. And what it is called integrated eicosanomics or integrated lipidomics. You look at the metabolic pathway, metabolic shunt, and look at the product, look at the substrate, and then also look at the gene and look at the expression. Now, going to the next stage, what Ed Dennis has done and his group have done, they looked at the individual product formation from these are all individual prostaglandins, these circles, and these rectangles are all the mRNAs. And they actually correlated with the time of the formation of the product from arachidonic acid upon cyclooxygenase or lipoxygenase action, and then also looked at the expression of the gene. And this is called integrated lipidomics or integrated eicosanomics. And this is very highly useful to look at stages of inflammation and also to target the condition of the pathology or disease state. Finally, what we are doing with Dr. Kraut and also Leslie Witkoff, who is actually helping us with a lot of things with the translational study on sickle cell disease. So we are looking at the sickle cell disease eicosanomics. We want to see what exactly happens in the serum, what happens in the blood cells. Can we actually combine these two and look at the eicosanomic profile both at acute and chronic inflammatory condition? One of the problems in sickle cell disease that we are looking at is acute chest syndrome. This happened in such a way, the collaboration thing, Dr. Kraut was looking at some help to do the SPLA2 analysis in sickle blood, because that was a marker for transfusion. So Dr. Clay March told up here he can do. So Dr. Kraut called and asked if he can do it. I said, well, we can do that. So the morning samples come, Leslie brings the samples, and then immediately we have to give the results by, within 1 hour. What happens here is that when we give the results, there is a level of SPLA2. What happens in sickle cell disease, nobody knows. When there is a chronic the acute symptom, the SPLA2, that secretory phospholipase 2, goes up 3 to 4-fold. And then they use it as a marker to do the blood transfusion. At a certain level, if it goes up, they actually do the blood transfusion. So we did that for about a year. And then we were interested to see. Then I talked to Dr. Kraut and Leslie, and I said, okay, why don't we look at this lipid profile and why this enzyme is activated, and what is the reason? So they said they actually put an IRB for us, and we have been working together now, and I will show you some of the data now on that project, what happens to these. So the real thing is we want to look at the lipid biomarkers in the acute phase as well as chronic phase, and then determine the COX and LOX metabolites to target that, and then also investigate where the SPLA2 comes from. We have an idea that it comes from macrophages, and then see what happens to the substrates like fatty acids. And finally, we're interested because there is a huge oxidative stress overload in sickle cell anemia because of the iron. So we want to see there's only one drug that is actually useful now is the hydroxyurea. And the hydroxyurea is also very highly used, and it's probably doing anything to the lipid metabolism or not, we don't know. There is also acute hemolysis in these patients. So when we looked at the LCMS profiles of these, we looked at the thromboxane B2 is very highly elevated. This is an inflammatory eicosanoid. And prostaglandin E3 is elevated. And a protective one, 6-keto-PGF1 alpha, is lower. That's actually, so what happens is the protective eicosanoids are down and the toxic eicosanoids are up. Similarly, the metabolites of lipoxygenases, the 9-heat, the 11-heat, and the 12-heat, they're up. So recently we found out that the 12-LOX is highly expressed. Actually, the activity of 12-LOX in the platelets of sickle cell patients is very high. So when I went back and talked to Dr. Kraut, he said, yes, we need to probably, platelet problem is a problem. So the other metabolites are also, they're changing patterns in those. Some are higher, some are lower. And then the hydroperoxy metabolites are high. So the heats also are higher in sickle cell patients. The fatty acid composition also changes. Some of the unsaturates like 18.2 and 18.1 are far down in the sickle cell patients. The polyunsaturated fatty acid levels are also down. 18.1 and 18.2 are down, and 20:3 is down, and especially the 20:5, the eicosapentaenoic acid is also down. The lipid peroxidation, actually, this is not a surprise. It's also up in these patients. So basically what happens is the lipidome profile in these patients having a chronic inflammatory response is the LOX and COX metabolites are up, and eicosanoids actually cause inflammation. On the other side, from the hemolysis, so there is an iron overload that causes oxidative stress and leads to reactive carbonyls, which also contribute to the inflammation. So the overall, there is the lipidome, proteome, and glycome, and the genome, and encircle, and actually they control the metabolome. That's what the final essence of this story. Thanks to my lab people and actually all my students to do this work, and I go and talk like this. Thank you very much for the opportunity.